Conformational dynamics of HBV capsid proteins in free and drug-bound states.
All right, so is this on? There we go. All right, so thanks everyone for sticking around. I know it's getting late in the evening. People are probably pretty tired, so hopefully I won't bore you too much. So I'm going to talk about some applications of methods, including some free energy, to a problem we've been working on now for a few years in my lab, specifically hepatitis B virus. All right, so just brief overview. So in the U.S., you may not think so much about hepatitis B, but it's actually a problem for at least 250 million people worldwide, maybe more. And in the U.S., around 1 to 2 million people suffer from chronic HBV infection. So it has no cure. You can basically just manage it, although some people have more symptoms than others. And there is a vaccine for it, and it has been for a while now, but obviously, not everybody, most people don't take it. It's not a commonly given one. And so if infected with HBV, it can cause liver cancer, cirrhosis of the liver, and eventually death. So it is a very problematic disease. So here's the life cycle of the HBV virus, or hepatitis B virus. So it comes, it comes into the cell. In a capsid, it is liberated, starts replicating its DNA, and then producing more virus. And so there have been over the years a number of attempts to interfere with this process. At various stages of the life cycle, but there's a major problem with them in that the biggest challenge is the persistence of this covalently closed circular DNA that remains in the cell even after after treatment with a number of different drugs. And so what we'd really like is a drug that not only deals with the symptoms, but ultimately eliminates even this persistent DNA, thereby curing the infection. Okay. So of course, so I do molecular dynamics simulations, so I think about things on the atomistic scale. So here is an example, a nice image from my colleague, actually Juan Perea at University of Delaware, of the hepatitis B virus capsid. It's about 40 nanometers, and it's composed, like all viruses, the capsid is composed of one or a few proteins, in this case just one protein called capsid protein, replicated 240 times, okay, or 120 dimers. So it looks something like this. Okay. So the dimer is the functional state. You really don't find monomers of this. And then from there, it interfaces with other capsid protein dimers at its edges. So those are highlighted and circled here. And so assembly proceeds through a number of intermediates. You have first dimers. They come together and can form tetramers, which then form hexamers. And then it's downhill from there to forming the capsid. Okay. And so it's this tetramer to hexamer, or somewhere in this early stage, that's the rate-limiting step for virus formation. Okay. So the assembly is believed to begin with weak energies. So for example, here's a dimer. So another dimer comes in and forms a tetramer, and the association of these two dimers is just a few kcal per mole. And it's believed to be weak to avoid kinetic traps. Whereas when you're going to the capsid, the last dimer to come in has more contacts and therefore more energy. And that's one of the reasons it's a downhill process. So in support of this, this idea of weak energies at these initial stages, if you make one mutation at an interfacial residue, so a tyrosine to an alanine, you don't completely break the association, so we still get, for example, tetramers forming, and even something kind of like a hexamer, but now it's a broken hexamer. So there's a gap where it should come together, and so from that point the capsid does not form. It will assemble into some other conformation. And so what this shows, though, is that this small interaction energy is normally needed, is small, but is sufficient to overcome a conformational barrier of sorts. And so when you lower that energy, you can't overcome that, and when you lower that binding energy, you can't overcome that conformational barrier. So in other words, a very simple way of looking at it is something like this, or the way I think about it at least, is that you have normally, the normal state of the dimer is a quote-unquote inactive one. Through, it explores conformational space in solution, and occasionally it happens upon a quote-unquote active form, where it can then bind to others more easily, forming the hexamer. And once it forms that, it's locked in, and then it's much easier for others to assemble on top of that nucleation point. So one question I had when I was first looking at this project was, could we understand what is the difference between active and inactive forms? I mean, it's just, in fact, when you compare structures, for example, one that comes from a crystal structure of a capsid versus one coming from a free dimer in solution, or one of these mutants that doesn't form capsids, the difference is really tiny. I mean, I look at these and I think, I can't imagine why one would be more accessible than the other, but in fact it is. And so I was trying to think of ways to get at this question. And so, I put this out there. This is an idea that we came up with, was to use a so-called eigenvector collective variable, which is essentially just a linear transformation from one conformation to the other. I calculated a free energy for this process and found, in fact, that, maybe you can say it's luck, but that the capsid-like state was higher in energy than the free state by about 5 kcal per mole or so. So, we're pursuing this a little farther now to see if this is really a reliable result, but it does seem to agree at least with the numbers estimated from experiments. Okay, so knowing that this conformational dynamics are important, the question comes up, can we somehow alter this dynamics to, say, change the pathway, the assembly pathway, misdirect it somehow? And of course the answer is yes. And there's a class of drugs called capsid assembly modifiers. So if this is just a couple tetramers that would normally assemble into a capsid, if you have these capsid assembly modifiers come in and bind, they can alter this pathway in various ways. They could inhibit assembly completely. They could misdirect it into non-capsid-like structures, or in fact, an interesting way they can modify this is to accelerate the assembly process, and the reason that that's beneficial is that then the capsid forms before the viral DNA is present. And so these classes have been known now for a few years. I don't know how helpful it is to show these, but this is one class, so-called capsid assembly accelerators. These are misdirectors, and they bind, if it wasn't clear from before, they bind at a pocket between the two dimers in the tetramer state. So thinking from this dynamics perspective, we want to know, can we figure out what distinguishes one class from the other? If just looking at how it interacts with the tetramer, can we already say if it's a misdirector or an accelerator? So to start, we of course have run some simulations. So this is, we ran 2 1 microsecond simulations of just a free tetramer to try to start understanding what sorts of conformational space does it explore when just left to its own devices. Okay. And so after a lot of trial and error, looking at different ways of quantifying this, we came up with 2 angles. So one between these so-called spikes. So when in the capsid state, they essentially stick out of the capsid. And then one that we call the base angle. between them. And so when we, so when we plot all the conformations observed in our simulation, so in this case, I'm only showing conformations observed in 2 times 150 nanosecond simulations, just for consistency with the rest of what I'm going to show you. We find that a tetramer samples a range of spike angles and particularly base angles, whereas if it's in a hexamer, it's locked in more tightly on that base angle. Okay. Now, because we got kind of tired of looking at just lots of dots, and it got a bit confusing, particularly when you have dots on top of dots on top of dots, my postdoc Anna came up with a better way of displaying this. So we use so-called standard deviation ellipses. So this essentially, it's drawn in such a way that it encapsulates 90% of the data points. So this is how I'm going to be showing it throughout the rest of this talk. Oh, sorry. Okay. So once we had that, those for the free state, we decided to simulate it with a lot of different known drugs bound. So in this graph, these are HAP compounds. They're known to misdirect the assembly away from the capsid into other unusual structures. And we found that the standard deviation ellipses all clustered down here. So basically at small spike angles, whereas the hexamer tends to have larger spike angles. So that seems to be telling us something, that the misdirectors are the drugs that misdirect assembly are causing even just the tetramer to explore a different conformational space. Now, for the accelerators, it's a little less clear. At least in one case, it very clearly overlaps with the hexamer-like state, so it's pushing the tetramer towards a state favorable to form a hexamer. And in the other one, it's a little less clear, but you can kind of see that it's at least constraining the conformational space, thereby possibly making it easier or faster to find a hexamer-compatible state. So now we want to say, can we use this way of classifying the conformational dynamics of the capsid protein in order to design completely novel drugs? So from our 2 times 1 microsecond simulations of the free tetramer, we picked a few states that fall away from the hexamer and somewhere in the center of the tetramer-like states. And from those 3, we carried out docking. So we also selected them such that they have a decent-sized pocket in order to be able to dock reasonably sized or even large drugs into it. So just a brief overview of what we did. So we screened 100,000 compounds from zinc and NCI databases, docking to our 3 structures from MD. We tried to exclude compounds that would already be unfavorable or toxic. And we landed on about 29 compounds ultimately that we had tested experimentally. The testing was done in the lab of our collaborator at Emory, so Ray Shinazi, and also working with Lita Basit and Brian Cox. So they were added to, so they have a very standard procedure for testing these. They added them at a concentration of 10 micromolar to these infected cells. And from those 29 initial compounds, 4 of them had some moderate inhibition. Okay. So that was okay. So we're thinking, all right, that's not too bad. But then of those, only 2 of them weren't toxic. So so that already ruled out half of our 4. So we're getting kind of nervous at this point. And then from that, 1 of them had demonstrated binding using trip fluorescence. So okay, so if that number, so this is where I was actually 2 years ago when I presented on this topic here at this meeting. If that were 0, my tenure in drug design would have been over about the time it started, but fortunately we had a starting point. So we've continued along these lines, and we've used this one compound to then look for similars and design a second round, second generation of compounds. So they're showing that not everything is just random, thankfully. From that, in our second round, we picked 19 compounds for experimental testing, and 7 of them had demonstrated inhibition in this Hep cell line. So at least 5 out of those 7 showed that they destabilized the virus through thermal shift assay, so lowering the melting temperature. And at least one of them so far has been tested using cryo-EM and shows that it alters the shape of the capsid or deforms the capsid. So that data is not ready for prime time yet, so I'm just showing as an example from this very nice paper last year. So this is the sorts of things that come out of E&M, so native capsids. Misdirectors, you get these very large or misshapen ones, and then accelerators, you just get, again, normal capsids, okay, but with no DNA. All right, so of course, we don't want to stop there, so now we're circling back around to the molecular dynamics, taking our compounds, simulating them bound to a tetramer, and seeing, okay, are our theories about how they classify into misdirectors or accelerators working? Do they make sense? So our initial compound, interestingly, has pretty good overlap actually with the hexamer state, but it wasn't very good at inhibiting, so maybe that's a sign why. And then our second generation compounds are actually starting to push further and further away from the hexamer, suggesting that maybe these are misdirectors. And that's in agreement with some of the experimental data coming out of the Shinazi lab. Okay. So of course, I didn't want to completely avoid FEP in this talk at this meeting. So we are starting to use FEP, which we'll need to do to optimize our lead compounds. So we wanted to test the methods, apply them to first known existing compounds. So starting from HAP12, so one of the misdirectors, we did a modification to HAP4. So experimentally, so this is estimated from EC50s, a ratio of EC50s, it's +3. With FEP, run by Chris Chapeau, we get 2.6. So okay, that's pretty good. We can further go to HAP1. Again, pretty good agreement, -1.6, -1. GLS4 is one of the better compounds out there in testing. Again, we get pretty good agreement. But then HAP7 went a little awry. So this one, from the EC50s at least, it's predicted to be greater than 4 kcal per mole, and we only get about +3. So, why is that? So we're digging into the simulations a little bit. When we look at the binding site, so here's the difference between HAP12 and HAP7, this group. I don't see, just by eye, I don't see why it should be so bad. And maybe this is a sign that just taking a ratio of the EC50s in some cases isn't always a good way of getting the delta-delta Gs. Okay. But maybe we can also blame the force field. That's an easy target. And that's definitely something we'll look into. We'll look into optimizing and see if that changes the results. Okay, so just wrapping up. So what I hope I've impressed upon you and what we've certainly learned, I don't have any experience any prior experience in this field, so we're learning that looking at the dynamics of the target, so not just binding free energies, but the dynamics can inform on how a drug works after it binds. And so we can classify them based on these 2 angles that we've defined. And that seems to correlate pretty well with the experimental classification into misdirectors and accelerators. So molecular dynamics plus docking has now given us a few new compounds to work on. So Again, not quite ready for prime time, so here's my schematic of one of our compounds. And then finally, that FEP should hope, at least in 3 out of 4 cases, we try to give really good sub-kcal per mole results. And so this should be useful for optimizing our compounds. And so with that, I'll thank my postdoc, Anna Pavlova, a bioinformatics master's student that really brought this project to me in the first place, Maxim Korobalov. He's now actually working here in Cambridge as a PhD student in an MIT Media Lab. My Emory collaborators, so in the lab of Ray Shinazi, also Lita Bassett and Brian Cox, and Chris Chapeau helped run some of the free energy calculations. And thank you guys.
Questions for JC?
Yeah. Next talk, could you say something about the similarity between the active compounds that you found and the one that you already know to target these interactions?
So to my...
Are compounds that you have been by anyway regarding the simulation that you have done?
So to my eye, I don't see the similarities. I'm not trained as a chemist. I'm trained as a physicist, so the chemicals all look pretty similar to me. So, you know, I think they're pretty different in any case, but we do need to dig in more deeply into the specific groups and interactions that are forming. Maybe the similarity is there. Yeah.
Could you probably have a charge change in your core-conforming FET perturbation?
Yeah, that's a good question, actually. That's something we should look at. Yeah. Yeah, I mean, literally, I was looking at this data yesterday, so in order to include it here. Yeah.
Any other questions? All right, let's thank JC again.